oxygen transport. How is oxygen carried throughout your whole body? That's what it comes down to. Once it gets into the bloodstream from the lungs, how does it go from one place to another? That's what this is. Okay? Well, 98% of it is going to be bound to hemoglobin, right? 4 molecules of oxygen go on 1 molecule of hemoglobin. And your red blood cells are loaded with hemoglobin. 2% is dissolved in plasma. There isn't much. Like I said before, oxygen is only 1/20th able to dissolve in water when you compare it to carbon dioxide. So you don't expect a lot to be in the plasma itself. Most of it will be bound in the red blood cells on hemoglobin. So hemoglobin, we have a heme group that has the iron in the center, okay? And we have 4 molecules of oxygen that bind to 1 hemoglobin. So we could have 4 on there. If 4 oxygens are on there, we say that the hemoglobin is saturated. All right, has 100% on it. If 1, 2, or 3 heme groups are bound to oxygen, then we say it's partially saturated. 25, 50, 75%. When we say it's partially saturated. Pretty easy to understand. Okay? That's what our heme looks like. All right? So that's hemoglobin. Here's the heme group with the center iron in there. Hemoglobin is made up of 4 chains. 1, 2, 3, 4. And in the center of each one of these chains, we have a heme group, the green thing. Oxygen combined to this, to this, to this, to this. And we have tons and tons of these hemoglobins in your red blood cells. Now here's some terminology. Oxyhemoglobin is when we have oxygen on the hemoglobin. And we refer to it as HbO2. You'll be seeing that quite often. Deoxyhemoglobin is when oxygen has been released off of hemoglobin, and we show it up like this, HHB. Okay? So the loading and unloading is a reversible reaction. Oxygen could come on here and knock off a hydrogen. Right? So now we got oxyhemoglobin here. Oxyhemoglobin. Hydrogen comes off. We're going to get back to that. Okay? Now, we have a curve here. Something called the oxygen dissociation curve. I know you guys are not big fans of curves, but this is one we can't get away from. And again, if you're going to respiratory therapy, it'll be your bread and butter. Oxygen dissociation curve. It shows the relationship of the percent of hemoglobin that's saturated plotted against the partial pressure of oxygen. So, we have this. This here will be the percentage of saturation. $25%, 50%, 75%, 100%. And this will be the partial pressure of oxygen. Okay, which would be millimeters of mercury. And we can still say 25, 50, 75, or 100 millimeters. All right? That's what this is, the oxygen dissociation curve. Now, it's not linear, it's sigmoidal. So let me explain to you what this is. If this is what linear would be. If my partial pressure of oxygen is 25, then that would say, if it's linear, then that would say that 25% would then be saturated of hemoglobin. All right? if I breathe in 50 millimeters of mercury of oxygen, then that will make 50% of my hemoglobin be saturated. 75 and 100. That would make for a linear curve, but that's not what it is. There's a neat phenomenon that happens instead. You need to understand this concept. Big concept. There's probably about a lot of questions on review questions and the percentage on the exam. This is what happens. If you put 25 millimeters of mercury that you breathe in, you've got to put a big pressure, you've got to start with a big pressure here. And that'll get you here. But if you do 50, you're not going to get 50. It actually is going to be much higher than that. See? Before it would be 50. It's not. If you put 75, it's going to be very high. Almost 100% saturated. And bringing it 100, it doesn't even do that much. So what I'm trying to show you here is this. That it's not linear, it's more of a sigmoidal curve. Where we're going to need a lot more pressure to get that first one on, but then the second and third one, it's pretty easy, and the last one, very easy. It makes it almost like S-shape. What I'm showing here is that even if you breathe in 75 millimeters of mercury, you don't have to breathe in 100 amount of oxygen. You just have to breathe in 75, and that's almost as close as making it go up to 100% of your hemoglobin filled. You see the concept there? You don't need to breathe in 100% of oxygen. Even at 20 millimeters of coming, you've got a lot of oxygen coming on. So there, in the middle portion here, it's steep. Okay? There's what we call cooperative binding. That once this gets bound over here, these two are much easier, and this one's very easy to log on. The best way I could explain it is think of a block of stamps over here. Back in the day when they would, well, I guess you could do it this way too. I remember licking them and stuff. But you have 4 stamps over here. Now I want to get them all off. So this is what happens. For me to take the first one off, I got to put more effort, more pressure to get the first one off. Because I have to break this bond, and I have to break this bond. And now I was able to do that. So the first one to get on, or the first one to get off, is going to be very hard to do. You're going to have to put more pressure on it. But because that first one came off, or on rather, that makes the other ones easier. It's cooperative. Hey, if you do the hard part, we'll be able to break off a little bit easier. Because look at this one. For me to take this one off, I only have to break off 1 bond. If I want to take this one off, I just have to break off 1 bond. This one comes off right easily. See how easy it is? There's no bonds even to break off. So that's what I'm saying here. You need more pressure to get the first one on, but the second and third, you don't need that much pressure to do. And the last one, there isn't much there to do at all. You're fully saturated almost with being just 75 taken in. You don't need to be breathing in 100 millimeters of mercury. I'm sorry, 100 millimeters of mercury of oxygen, though. Does that make sense? Can you visualize that? All right? So when the first oxygen binds to the oxygen binding site, the 3 other sites bind quicker. Cooperative binding. All right? There's the first one. But look, you have to put, there's the first one. See, it's only 1 on here. But you've got to put more pressure to get the first one on. second and third one, not too bad, and the last one, pretty easy. All right? This is what it really looks like. I kind of drew it to scale, but this is what it really looks like. So look what's happening here. They put numbers here so you can see it. Here you are at 20 millimeters of mercury. Sure, it'll be about 25% over here. Well, here, they're saying, oh, you can just look over here. 20, I'm not really good, so 35. So at 20 millimeters of mercury of oxygen, partial pressure of oxygen, you have to put more pressure to get that first one on. Does that make sense? Okay? But look, the second and the third one, it's not too bad. What I want to say over here is like, even if the PO2 is at 100, well here, look at this over here. At 75 millimeters of mercury, 75, you're at, what, about 93% saturated? See, here's 70s, 92, 80s, 94. So as you get up here higher, you don't need to be breathing in a whole lot of oxygen. Our bodies have adapted that we've been under less oxygen, our hemoglobin could attach to it, or the oxygen can attach to the hemoglobin. At 75, it's like 93%. So even if you want to raise up the amount of oxygen that you're going to breathe in to 100%, you're going from 93 to, it never really makes it up to 100%, but it goes like 97.5%, which is pretty close. But there isn't much of a difference by just bringing it up to 25. Does that make sense? So if the PO2 is at 40, the hemoglobin is at 75. At 40, at low levels of oxygen, you're 3/4 saturated. That's amazing. I'm trying to not inspire, but I'm trying to show you the significance for this and how much there's going to be questions on this. You see that? At 40 millimeters of mercury, you're 75% saturated. You don't need high levels of oxygen to be breathing in. We do well with low. Even at 75 millimeters of oxygen, you're at 93%. To go 93% to 100, you have to put another 20. You don't need to do that. You're already pretty much up there. Do you see the significance? Okay?
What kind of questions do you have?
Oh, look at my review questions. You'll see it. Because then we have to apply this oxygen dissociation curve. What happens when the pH changes? What happens when the temperature changes? What happens in the amount of carbon dioxide? All these different things, which we'll do next week, to get heavier. We've got to apply this. Okay? Yeah. All right? So that's what's happening over here. All right? So hemoglobin is almost completely saturated with a partial pressure of oxygen at 75 millimeters of mercury. You don't need 100 millimeters of mercury. You don't, when people are breathing with oxygen tanks, you don't need to bring it up to 100. Do you see what I'm saying? They do well with lower amounts. Because it gets capped off. If they're not doing well at 75, at 93%, they need something else going. There's something else going on. Okay? So further increases only produce small increases in oxygen binding. At 75, you want to bring it up to 80, it's only going to do a little bit. It goes from 93 to 92%, or 93 to 94%, not much. You don't need that much, is what I'm saying when you get up there. Oxygen loading delivery to tissues is adequate when partial pressure of oxygen is at, are below normal levels. You don't need to be bringing up high levels there. We're creatures that do well with low levels of oxygen. Okay? Only 20 to 25% of bound oxygen gets loaded in one systemic circulation. Meaning, at rest, now look what's going to happen here. Red blood cell comes up. It's going to get saturated with oxygen, 100% saturation. It's going to go around your whole body. By the time it gets back here, 75 is still on it. Only 25% came off. In a normal, healthy person at rest, what you're doing right now. So what's the other 75% on there for? You ever hold your breath and you're not taking in oxygen? So how are you breathing then? How are you living? Because you have other 75% on there. See how you hold your breath? There's going to be a point when it's going to go down like 25%. And you can't help but take a deep breath. But you have 75%, hint, hint, 75% on reserve on your red blood cell. That's at rest. You're running a marathon, you're not going to be able to do that. Because your body needs a lot more oxygen. So if you're running a marathon, when it comes back up here, maybe 40% is on there. Because we used a lot more because your body needs more oxygen. Okay? So if oxygen levels in tissues drop, more oxygen dissociates from the hemoglobin and is used by the cells. If you're running a marathon, more oxygen needs to come off of that. Okay? So you don't need to increase your respiratory rate because you have that reserve. You have that 75% that's still on there. Sure, if you start using it all up, you're going to have to increase your respiratory rate. You're going to have to increase your cardiac output. But you don't need to at rest. Okay? So the nearly complete saturation of hemoglobin in arterial blood explains why breathing deeply causes little increase in O2 saturation. You don't need to at rest because there's all that 75% that's still on reserve. Okay? And that's what the whole oxygen dissociation curve is. All right? At 40, it's at 75% saturated. That's pretty damn good. Really good. Okay?
All cells need oxygen. It is the essential fuel which is necessary to enable cells to stay alive and to carry out their various activities. Bringing oxygen to the cells requires the uptake of oxygen from the air in the lungs, its transportation in the blood, and its delivery to cells all over the body. The first step is the taking up of oxygen by blood flowing through fine capillaries in the walls of the lungs' air sacs, or alveoli. The oxygen molecules change from their state as a gas freely circulating in the air, dissolving into a solution in the plasma within the capillaries of the alveoli. Once in the solution of the blood, 98% of this dissolved oxygen is taken up by passing red cells, leaving just 2% remaining in the physical solution unattached. Red cells are particularly well suited to transporting oxygen because they contain a special oxygen-binding protein known as hemoglobin. Each molecule of hemoglobin itself contains 4 molecules of heme, an iron-containing pigment, which binds oxygen loosely and reversibly. Hemoglobin that is fully saturated with oxygen is bright red and is called oxyhemoglobin. On the other hand, hemoglobin that is not saturated with oxygen is purplish-blue in color and is called deoxyhemoglobin. It is heme which makes it possible for the red cells to pick up oxygen dissolved in the blood, transport it combined with hemoglobin, and release it back into the blood as oxygen in solution, ready for delivery to the various cells of the body. Hemoglobin gives up its oxygen as red cells travel through capillaries in tissues where there is a low content or partial pressure of oxygen. The partial pressure of oxygen represents the level of dissolved oxygen in plasma. As oxygen is released and again is carried in solution, the partial pressure of oxygen in the capillaries becomes greater than the partial pressure of oxygen in the surrounding tissues. This causes oxygen to move out of the capillaries into the tissues and to finally reach the cells. This graph, the oxygen dissociation curve of hemoglobin, shows why hemoglobin is particularly suited to its role in transporting oxygen. The oxygen dissociation curve demonstrates the relationship between the oxygen carried in combination with hemoglobin, the O2 saturation, and the partial pressure of oxygen in the arterial blood. The sharp upstroke and the flat plateau illustrate how oxygen is released to the tissues over a wide range of conditions. Its shape means that although the partial pressure of oxygen in the blood returning from the lungs and being pumped out by the arteries may be reduced to only 50% of the normal value, say due to lung disease or high altitude, hemoglobin will still be 85% saturated with oxygen.
Welcome to the first episode of Talking Graphs. What this series of weekly videos is going to be talking about is it's going to be looking at different physiologic relationships and look at how they can be illustrated using a graph. So for anyone studying or learning about physiology, you'll notice that graphs are used all of the time to look at the relationship between two variables. And they're incredibly valuable because a graph itself, if you don't get lost in all the details, if you just step back and look at a graph, it's actually a symbol of how an enzyme or a molecule or some entire organism behaves. And so when we look at the hemoglobin and oxygen curve that we're seeing here, you have two variables. On the x-axis, you have partial pressure of oxygen. Now, most of the time when they illustrate this graph, they don't tell you exactly where that partial pressure is because it could be in the lungs, could be in the active tissues, and so they leave off any location for the PO2 because it can apply to more than one location. But for the point of view of, or for the benefit of learning this for the first time, I want you to imagine that that PO2 is in the tissues of the body. So the 100 over here is 100 millimeters of mercury, which is the pressure of oxygen, but you can just think of it as a lot of oxygen. And it doesn't mean 100%, but you might as well think of it that. It means a lot of oxygen over here, very low oxygen over here. And again, think of this as the tissues. I think it's useful for learning this for the first time. And then over here on the y-axis, you have oxyhemoglobin percent saturation. And so in this case, it is a percent. And 100 represents 100% saturation of the hemoglobin. So that means the hemoglobin's holding as many oxygens as it possibly can. And then down here, it's as little as it can. And so normally, hemoglobin, when it's in an environment where you have between about 190 millimeters of mercury of oxygen around the hemoglobin, so this would be in a tissue, for instance, again, we're going to look at it from the tissue perspective. This would be a tissue that is not doing much. And the tissue has lots of oxygen present, it's not very active, and so the hemoglobin that passes by that tissue is going to hold on to all of its oxygen molecules, and it's going to continue on to somewhere else, maybe where the oxygen level is lower. And so this curve for hemoglobin is what's called a sigmoid curve. It just means that it's an S shape. And this top region, this area that's more flat, has the least slope, that area is where hemoglobin has a very high affinity for oxygen, and that means that it holds on to it very tightly. And you can see that it holds on to it tightly because when you look at the x-axis of this graph, between about 170, even though you have a big change in the partial pressure or the amount of oxygen around the hemoglobin, you have a very small change in the percent saturation, which means that hemoglobin is not giving away the oxygen. It's holding on to it or has a very high affinity. But then this S-shaped curve goes into a very steep downhill area. This steep area is where hemoglobin starts to behave a little differently. When hemoglobin gets into environments where the oxygen level is below about 60 millimeters of mercury, so now we're getting into some active tissues that are pushing down the amount of oxygen because they're consuming it. And so when you get into this range right in here, now hemoglobin behaves differently. Now for a very small change, let's look at between 50 and 40 millimeters of mercury, you get a very large change in the amount of oxygen that's bound to hemoglobin. It goes down, meaning that the hemoglobin is giving off oxygen to those active tissues. And then as you go even lower here from 40 to 30, here's 40 on the line, here's 30 on the line, huge change. So a very steep part of the line means that the hemoglobin is dropping in saturation very rapidly. So these are going to be areas, again, where the tissue, PO2, is very, very low. And it's going to continue down this very steep curve. And the reason that this steep curve is very valuable is that hemoglobin is only giving off a ton of oxygen in areas where the PO2 is very, very low. And this ensures that the hemoglobin will deliver oxygen to the regions that need it most, rather than give it all away to some area that's only slightly active or something like that. So it holds on to it until it gets to tissues that are very, very low in oxygen. And that overall is kind of the big picture here. So I'm going to do a second video that talks about these variables here. But overall, just to sum it up, basically hemoglobin holds on to oxygen very tightly until it encounters a region of your body where the PO2, or the amount of oxygen, is extremely low. Then it changes its behavior. It gives off those oxygens very quickly. And again, that relationship is symbolized by the shape of this curve.